Hello and welcome to Science Research Weekly, Season 4, Episode 11, Dense Subject Matter. Get ready for cell cycle, synced endosymbionts, cytoskeleton densities via deep learning, taking the sting out of autoimmune diseases, constraints on the internal matter of neutron stars, and a protein association grant for the American Heart Association. This episode covers material from March 14th to March 20th. Let's get started with our research articles. First up, a nucleus-encoded dynamin-like protein controls endosymbiont division in the trypanosomatid Angomonas deanei. Stay with me here on this. It's complex. Kind of is, but the big picture is really exciting. So, eukaryotic cells have organelles like mitochondria and chloroplasts that used to be free-living bacteria, so endosymbionts. This happened 1.5 to 2 billion years ago. But some eukaryotes have other endosymbionts of a more recent origin, which are much more like bacteria that are just still inside them rather than developing into full-blown organelles. Anyways, Angomonas denai is a single-cell eukaryotic protist, for lack of a better term. It has a beta proteobacteria endosymbiont, a kinetoplastic bacteria, that arose a mere 40 to 120 million years ago, provides essential metabolites to its hosts. So researchers found a protein, ETP9, in the protist that essentially spurs on the cell division process of kinetoplastic bacteria so that the cell cycles of both the protist and the symbiont bacterium are synchronized. So that's really interesting, seeing kind of the development of how endosymbiont becomes essentially an organelle. And that has reflections into how this happened farther back in the past with things like mitochondria and chloroplast. All right, with that, let's turn to our next article, deep learning-based cytoskeleton segmentation for accurate high-throughput measurement of cytoskeleton density. So researchers were able to use deep learning on cytoskeleton microscopy images to obtain accurate quantitative measurements of density, which is really helpful in microscopy work, especially in like plant cell biology. And I think outside of deep learning, it's doable, but it's really intensive. So this really helps streamline the process. All right, our third article, ARF-GAP2 promotes STING proton channel activity, cytokine transit, and autoinflammation. So autoimmune diseases occurs, sort of simplified, they occur when cells in the body respond to an immune threat that isn't there, stressing out and damaging the body, cells in the body for no reason. So researchers working with a rare disease called STING-associated vasculopathy with onset in infancy, or SAVI, found that STING, so this is stimulator of interferon genes, it's a protein, works with another protein, ARF-GAP2, so which means associating with the Golgi. This protein is associated with the Golgi apparatus, and I think it helps actually acidify the Golgi apparatus, which is important for proper protein development. Anyways, so it works with STING and ARF-GAP2 work together, and they actually contribute in this disorder to autoinflammatory disease pathogenesis, and that actually simply turning on ARF-GAP2 could turn off or turn down the autoimmune attack. So really show that so like mice without this gene had improved lung disease. So really interesting thing here of ways to help modulate autoimmune diseases by understanding more about pathways and how it actually happens in the first place. All right, we'll close out. We did a lot of cell biology, so let's close out with some astrophysics. So the final one is QCD, constraints on isospin-dense matter and the nuclear equation of state. So like all good astrophysics paper, the title gives no clue for a non-specialized reader on what it's actually about. So essentially, from what I understand here, physicists modeled the internal structure of the matter of neutron stars and the QCD and the title means it's the abbreviation for quantum chromodynamics. So, yeah, from the title, you have no idea that it's about neutron stars. Obviously, very dense physics equation, but really trying to figure out more of how that incredibly dense matter in a neutron star actually works and how we can understand it. So, with using quantum chromodynamics, which sounds like a great name for a Magic the Gathering card, if you ask me. All right, we'll close out our article time here and get to some research tools. So no new R-bloggers posts of note or other statistical tidbits, so I'll go right into new cram packages that might sate your statistical appetites. There is Globsco Swarm Intelligence Optimization, Minimalist GODB, Build a Minimalist Gene Ontology Database, Chi-square, visualize contingency tables using chi-square plots, and snow data. Historical data from Jon Snow's 1854 Chloria outbreak map. And closing out our time here, some funding from the American Heart Association has pilot grants to explore the use of the American Heart Association protein binding atlas to discover novel protein interactions. So these are projects aimed at predicting and understand protein-protein and protein-small molecule interactions involving key biological processes associated with cardiovascular, kidney, and metabolic diseases, including obesity. Unsurprisingly, the proposed projects are encouraged to utilize the American Heart Association protein portal. What is the hadronic matter with you? Thanks for joining me for Science Research Weekly. Science on.